joining us for the CGW 6.0 overview webinar. Today we'll be exploring the new aggregated data and expanded constitutional testing features that are part of CGW 6.0. I wanted to take a moment to also say thank you to everyone that's been using CGW and is a part of our partner sharing network. Our hope is that as we expand CGW, it continues to be the one space and full solution for your NGS workflow. This update and all updates come directly from feedback from our partner sharing network, and we use this feedback to continually improve our software and make it better for the end user. We'll be taking questions at the end of this presentation, so please use the questions window in the GoToWebinar control panel to send questions as you think of them, but we'll be addressing them at the end of the talk. I also wanted to make a couple of announcements and make you aware of a few upcoming events in the next few months. First, we will be at ACMG from March 22nd to 24th in Phoenix. Please visit us at booth 511, where we can give you a hands-on demo of CGW 6.0 and also give you insight on what is coming later in 2017 from PierianDx. Second, also in March, we will have a webinar that's presented by one of our CGW network partners, the University of Vermont Medical Center. Dr. Nikki Sideropoulos will be the presenter, and she will be discussing insights that she has based on her experience in streamlining their NGS program. This webinar will be, like always, the second Tuesday of the month, which is March 9th, also at 11 a.m. Central Time. So with that, I'll move on to introducing our speaker for today, Dr. Bryce Daines. Bryce is PierianDx VP of Product Management. He joined us from Tooth Genomics, which PierianDx acquired a few months ago, where he served as Vice President of Product. He obtained his bachelor's degree in bioinformatics from Brigham Young University and a PhD in human genetics from Baylor College of Medicine, where he trained at the Human Genome Sequencing Center. Bryce has a lot of experience in next-gen sequencing and analyzing whole genome, exome, transcriptome data, also in the development of bioinformatics pipelines for both Illumina and IronTorrent platforms. He serves also as I introduced the VP of Product Management at Pierian. So I'll hand it over to you, Bryce.
Welcome. Thank you for joining us. Today we'll be providing an overview of new features in the latest release of CGW, version 6. This release of CGW is now available to users within the CGW test environment. This release contains several new features, including a clinical data aggregation feature, which I will discuss and demonstrate today. Clinical data aggregation leverages the breadth of the PierianDx Partner Sharing Network. It enables the display of aggregate variant frequency and classification across institutions, information which can be useful for informing classification decisions. I'll also demonstrate several new features supporting large constitutional panels and whole exomes, including phenotype-based prioritization, new filtering capabilities, and variant classification following the ACMG 2015 guidelines. The features presented in this webinar will be available in production environments beginning March 10th. CGW version 6 will include a new feature leveraging clinical data aggregation across the PierianDx Partner Sharing Network. The Variant Details page now includes a Clinical Data Aggregation tab. This tab includes two views of aggregated data. The first is a view of data aggregated at the variant level, which provides a count of the variant across disease indication and classification. The second is a view of data aggregated at the gene level, which provides a count of variants within the gene across 4 features: variant type, classification, consequence, and functional consequence. Clinical data aggregation is a new CGW feature available in this release. The Clinical Data Aggregation tab can be found by navigating to the Variant Details page and clicking on Clinical Data Aggregation. The Variant Level Frequency section contains two charts. One, the frequency of the variant by disease, and the second, the frequency of the variant by classification. To add additional diseases to the variant frequency by disease chart, click on the small plus icon next to the term Add Disease. This will open the Select Term dialog. Select the term from the drop-down and confirm the selection from the tree. Here we can see the frequency of this variant, PQ61K in NRAS, within malignant neoplasm of colon is more common than in malignant neoplasm of large intestine. In both cases, this variant is commonly classified as a level 2 variant. The frequency of this variant can be viewed by hovering over the bar, in which case CGW indicates that this variant has been observed in 102 out of 204 relevant cases. The numerator represents the subset of cases for which the variant was called and the variant passed the appropriate quality filters. The denominator represents the number of variants for which the NRAS gene is present within the gene list tested by the assay. The gene variant frequency display includes 4 graphs: the variant type, classification, consequence, and functional consequence of variants found within the NRAS gene are displayed here. The user can adjust which variant classifications are included in displays, for example, by removing level 4 and level 3 variants. Here we see that the majority of level 1 and 2 variants are single nucleotide variants whose consequence is missense and whose functional consequence is predicted by SIFT to be deleterious. We can adjust the functional consequence scores displayed in the chart by selecting other algorithms from the drop-down list. CGW now includes support for multi-specimen procedures for selected panels. Some changes to the core workflow have been made in order to accommodate procedures that involve multiple specimens. These include changes to the procedure for accessioning cases with a multi-specimen scheme, changes to the sequencing tab to support accessioning of one or more sets of sequencing details per specimen, changes to the Analyze tab to support launching of informatics jobs for each specimen, and finally in reporting. The report is generated for only the primary specimen, and by default the primary specimen variants are displayed on the Variants tab. However, the user may view variants called within secondary specimens. Here on the Accessioning tab, when creating a new case, in order to enable a multi-specimen panel, specimen details and patient details must be provided for each sample. Here's an example of a trio workflow, which includes specimen details for the proband, or primary sample, and 2 other specimens, in this case the mother and father. Changes have also been made to the Sequence tab. Users must now add sequencing run details for each specimen in a multi-specimen workflow. Additionally, the Analyze tab will now include sequencer run details for all of the specimens included in a multi-specimen panel, as will the run details and analysis files under the informatics jobs contain one set of records for each specimen included in a multi-specimen panel. Users now have the option to manage their own password using the Change Password dialog. Please note that for compliance with regulatory requirements, strong passwords are now enforced by CGW. Password self-service allows CGW users to change their own password. Start by logging into CGW. From the upper right-hand corner, hover over your username. From the drop-down menu, select Change Password. This opens the Change Password page. Provide your current password, followed by your desired password. Please note that passwords must conform to the password complexity policy outlined here. This includes at least one uppercase letter A through Z, one lowercase letter A through Z, a digit 0 through 9, and a non-alphanumeric character. CGW version 6 includes significant enhancement to features supporting constitutional panels and whole exome analysis. These features include phenotype-based prioritization of variants with Phenolyzer, rapid classification of germline variants following the ACMG 2015 guidelines, and expanded filtering capabilities, including the ability to filter by genotype and mode of inheritance. Phenotype-based prioritization with Phenolyzer enables more efficient review of complex constitutional panels or whole exome sequencing cases. Phenotypic terms are provided for use by Phenolyzer when accessioning cases for which phenotype-based prioritization has been enabled. Phenolyzer takes as input terms phenotypic descriptions from the human phenotype ontology and calculates a score for each gene weighted by the strength of association between the gene and provided phenotypic terms. CGW now includes 3 new data columns: Phenolyzer disease, Phenolyzer terms, and Phenolyzer score, which are available in the Variants tab. Now I will explore the variants in the hemolytic anemia trio on the Variants tab. Here I'm already applying a filter based on the consequence of the variant, looking for those that may be altering the protein, and also based on the frequency of the variants in the NHLBI population. Now we'll add a filtering step based on the Phenolyzer score. The Phenolyzer score ranges from 0 to 1 for variants and genes positively associated with the provided phenotypes. I will apply those changes, clicking here. And now I have a reduced set of results. We've gone from the 35,000 variants in the proband down to just 86 possible variants. 3 Phenolyzer-associated columns have been added to the Variant tab: the Phenolyzer diseases, Phenolyzer score, and Phenolyzer terms. The Phenolyzer terms are the list of terms accessioned for the case that match to the gene or variant represented in this row. The Phenolyzer score is a weighted score calculated by Phenolyzer based on the prevalence of associated terms within the Phenolyzer database and the strength of association between the gene and the terms accessioned for the case. Finally, the Phenolyzer diseases are a set of potential diseases inferred based on the provided phenotypes. Here I'll sort the results based on the Phenolyzer score. Sorting the results based on the Phenolyzer score puts the most relevant candidate genes and variants at the top of the list of results. In this case, the causative gene, or the causative variants, in the gene PKLR are represented in the top 2 results based on Phenolyzer and the filtering criteria that we've applied above. The Variant Details page now includes an ACMG Evidence tab displayed for panels that use ACMG-based classification. This tab displays all the standard ACMG evidence codes with 3 selectable options, yes, no, and not evaluated, which indicate whether an individual evidence code has been inferred. CGW will auto-populate many of the ACMG evidence codes based on supporting evidence inferred from the CGW knowledge base. The ACMG Evidence tab provides a guided workflow for calculating a classification. Updating a report with an ACMG guided classification is a separate action, which I will demonstrate. From the Variants tab, click on the variant name and open the Variant Details page. On the Variant Details page, you'll see a new tab, ACMG Classification. Navigating to this tab will open the ACMG classification dialog. You'll find that some of the evidence codes for the ACMG classification are automatically inferred by CGW based on the evidence available in the Variant Details page. For example, this variant has been inferred as having a PS2 because it is a de novo variant based on the confirmation sequencing in parental samples. You'll also see that many of the evidence codes are not yet evaluated by CGW's algorithm, but we plan to expand on the capabilities of automatic inference in subsequent releases. When you navigate to the ACMG classification page, an initial or calculated classification is indicated here, in this case, likely pathogenic. You can also see the current classification as provided in the report, in this case, a classification of level 5. The evidence codes supporting the ACMG classification can be provided by the user where they are not automatically inferred by CGW, or the user may override the automatic inference. For example, this variant has been detected in trans with a known pathogenic variant in an individual that caused disease. Therefore, I can indicate that the PM3 value is yes and recalculate or update the classification by clicking here on Calculate Classification. To update the classification of this variant in the report, click on the Update Variant Classification and Report button. You'll see this change reflected above in the current classification and report, now level 2 or likely pathogenic. CGW has enhanced filtering capabilities, including 2 new sets of filters, the genotype filters, which are available for all panels, and the mode of inheritance filters, which are available only for select constitutional panels. Additionally, an important change in functionality should be noted by CGW users. Changes to the filtering criteria are no longer immediately applied when selected. In order to apply changes to a filter, users must click the Apply Changed Filters button, which appears when filter criteria are modified. Case review for large constitutional or somatic panels, including whole exomes, can often involve frequent changes to the filtering criteria. In support of these more complex workflows, we made a small change to the filtering dialog. When making changes to the filtering criteria, such as altering the consequence to include non-synonymous mutations, the changes are not immediately reflected in the data table below. In order to apply the changed filter to the data table below, click on the button Apply Changed Filters. You'll notice that this appears whenever the criteria for filtering are altered and goes away when the filter has been updated. Complex panels and whole exome sequencing can generate large numbers of variants. For example, the case I'm reviewing here of a hemolytic anemia trio used whole exome sequencing and generated more than 35,000 variants. Filtering variants from these cases can be complex. To enable more efficient case review, we've added several new filters to select panels. These new filters can be found on the Variants tab in the Filtering dialog under the Other Filters menu. By expanding that menu, you can now see the genotype filters and inheritance model filters. The genotype filters will return variants based on the zygosity in the case. For example, the heterozygous in case filter will return variants which are heterozygous in the case or proband and either homozygous reference, homozygous alternate, or missing for all controls, or the parental samples. To see the logic applied to genotype or inheritance model filters, just click on the small letter I next to the filter name. The inheritance model filters are useful for filtering variants which fit a known or suspected mode of Mendelian inheritance. For example, the compound heterozygous filter returns all variants which satisfy the following conditions. There are 2 variants present in the gene after all other filters are applied. The offspring is heterozygous for both variants. And each variant is present in exactly one parent. As a final note, in this version of CGW, we removed the archiving feature, which had previously archived samples after a certain period of time post-sample accession and report generation. This means that users will no longer be required to archive or unarchive their cases in order to use all features of the platform. The actions and buttons associated with case archiving and unarchiving have been removed. With that, we'd like to thank you for your time and attention on our webinar today as we presented you the new features of CGW version 6. At this time, we'd like to take your questions. You can provide those questions via chat, and we will answer them now.
Thank you, Bryce. So there is a questions window where you can send any questions that you have. We'll get started with the few that we have. The first question is whether the frequency by classification in the clinical aggregation feature that Bryce described, whether that includes all reports or whether it or whether it just includes published interpretations.
Right. So the frequency by classification feature, and in fact all of the frequency filters or frequency features that we display, the data aggregation features, are aggregating data across all cases which have been signed out, or all reported cases, not just those that are published interpretations.
Okay, we'll move on to the next question, which is whether the Phenolyzer terms can be passed via an interface. So I'm assuming that means HL7 messaging, things of that nature, or API.
Right. I'm not sure if the question is asking about an API or whether it's asking about the user interface. In both cases, we've had support for accessioning. Thanks. There's clarification on the line now, API. We've had support for accessioning phenotypes within the user interface and the API for phenotype-based workflows in the past, but we continue to support those via the same mechanism.
Yes, so that is for API support as well as HL7.
Right.
Okay, the next question is, with multiple specimens per case, do you support tumor and normal with appropriate filtering? So,
right, so for instance, in a tumor normal paired approach to somatic testing, we would probably work with you to develop a pipeline that would do a subtractive analysis to remove germline variants. But you can then also view those germline variants. You can toggle between specimens in the variants tab. So there would be ways to build a pipeline and your panel in order to show just the somatic or show both as you desire for that type of assay.
So there's a question asking for some clarification on whether there is a sample size, so the number of reports or cases that are used to arrive at the variant frequency.
Okay, so as we demonstrated, and we can pull this up again.
Share your screen.
Let me share my screen so I can demonstrate this briefly again. As we showed on the clinical data aggregation tab, it was probably a little bit quick when we presented it previously. On the mouse over of the appropriate bar here in the variant frequency by disease or in the classification charts, we can actually see the numerator and denominator that are contributing to the calculation of the frequency. So again, in this case, it's indicating 115 observations of this variant out of 375 reported cases of malignant melanoma.
This also highlights then the fact that you can see the frequency within your institution in blue and then across all Pierian network members in red. So you can gauge perhaps whether you're seeing the frequency similar to what others are seeing and then get the benefit of the full set of cases across that have been run in CGW for that disease.
Or also maybe we can add how this is telling us how others are classifying.
Right. So this pair of red and blue in the left graph also indicates really how we show all institutions on the top and my institution at the bottom. So this pair of bars here will show you that you can see 6 out of 6 cases at my institution were classified as level 1, is indicated in red here, and then 117 out of 117 at other institutions. And then in some other diseases, you find that it's not always classified as 1. In glioma, for instance, it's sometimes classified as level 2. And so this can help you gauge what others who are sending out cases in CGW have determined as the appropriate classification level for that disease.
Yeah, so I think there's another question, Bryce, that is about accessing archived data. So perhaps we can clarify how the analysis files would now show up at all times. So you guys are probably familiar as users of the Analyze tab within CGW. And this tab shows various data files that are generated as part of the CGW analysis. That could be the BAM file, the VCF file. Previously, these files would be archived 2 weeks after report generation. And this no longer happens. So the files are always available. There is no archiving that occurs anymore. So you'll always be able to see them. Do we have any?
And I think importantly, their previously archived cases will be unarchived by default, and there will be no longer an archiving action or an automated archiving.
So I think that wraps up our questions for today. Thank you everyone for joining, and you can look out for the recording of this webinar to be sent out to you shortly. And please join us next month for the webinar presented by one of our CGW users, Nikki Sideropoulos from the University of Vermont Medical Center. Thank you and have a good rest of the week.